thank you very much for the invitation. And so I will come back to gene editing and specifically CRISPR-Cas9. To start, I will want to give a summary for the community. So the question we are trying to address is whether CRISPR-Cas9 treated blood stem cells can persist long-term after they are transplanted. And of course, we are questioning whether this approach is safe. The findings that I'm going to show you describe how those edited cells can persist for over 2 years at high levels with no adverse side effects in non-human primates. This is important because this approach is potentially applicable to many genetic and infectious diseases such as HIV. And we are excited about this because, as we have heard this morning, this type of approach is currently being tested in patients with HIV, but also with hemoglobin disorders, which is what I will focus on. And hopefully, this approach will also become available for other diseases. So I will talk about the use of CRISPR-Cas9 editing for the treatment of hemoglobin disorder, such as sickle cell disease. And instead of trying to correct the mutation that causes the disease, what we have tried to do is actually to reactivate fetal hemoglobin, because fetal hemoglobin has been known for a few decades now to ameliorate the symptoms of patients that suffer from hemoglobinopathies. And this is illustrated in this graph where you can see increased survival in sickle cell patients that express high level of fetal hemoglobin. Now, in most adults, you and me, fetal hemoglobin is actually silenced after birth. And this is illustrated in this diagram where you can see the downregulation of the gamma globin gene, which is a component of fetal hemoglobin, and replacement with beta globin expression to make adult hemoglobin. So what we are doing is actually to focus on the promoter region of this gamma globin gene to try to mutate a known binding site for a protein called BCL11A, which is a repressor of gamma globin expression. And so essentially what we have done is to use a CRISPR-Cas9 site in this region to target this BCL11A binding site. And we are taking advantage of the non-homologous end joining pathway that Paula Cannon described this morning, which essentially insert small insertion of deletions at the cut sites. We are using this pathway to actually mutate the BCL11A binding sites to now increase expression of those gamma globin genes. And I want to emphasize that even though we are interested in the effect on red blood cells by looking at hemoglobin production, we are actually doing the editing at the level of blood stem cells because we are looking for a long-lasting effect, and we know that those stem cells can self-renew. So specifically for our studies, we were interested to evaluate the engraftment of those CRISPR-Cas9 treated blood stem cells, and we have used a non-human primate model to address this question. So this is our standard transplantation protocol that we use in the lab, where we are first purifying CD34 positive cells from the bone marrow of these animals. And on day 2, we are treating these cells with CRISPR-Cas9 ribonucleoprotein electroporation. And those edited cells are now infused back in the animal that receives myeloablative conditioning to allow those cells to engraft. And as far as measurement goes, we were specifically interested in following engraftment as well as in vivo editing efficiency. And of course, we were interested to see if fetal hemoglobin was reactivated in those animals. So we started with a first cohort of 3 rhesus macaques, and I wanted to emphasize the number of cells that we had to edit for this transplantation experiment. As you can see, we had to edit between 80 and 200 million CD34 cells for each animal, which is quite a bit of cells to manipulate. And the results of the in vivo editing is shown in this graph for the 3 animals. What you can see is we were quite excited to see editing levels that stabilized at pretty substantial level that range from about 10% to 25% in the 3 different animals. And this was the case for almost 2 years post-transplantation. Since we use deep sequencing to measure editing efficiency, we were also able to look at the different deletions that were generated in these animals. And this is shown in the form of the bar graph where each deletion is color-coded. And you can see down below here that most of the deletions that were generated from the CRISPR-Cas9 editing ranged in size from 1 nucleotide to 30 nucleotides. And importantly, all of these deletions overlapped with the binding site for BCL11A and are therefore expected to interfere with the activity of this protein. Now, we were excited with this result, but as I mentioned earlier, the main challenge was the manipulation and the editing of such a large number of CD34 cells. And so to circumvent this challenge, we wanted to test a new refined phenotype for hematopoietic stem cells that was recently described in our group, and that is mainly based on the marker CD90. So if you look at the bulk CD34 cells, the CD90 positive expressing cells only consist of about 10% of those CD34 cells. And so in a new transplantation experiment, what we have done is first sort out the CD90 positive cell from the CD90 negative, and we only edited this CD90 negative fraction, but at the time of the transplantation, we combined those edited CD90 positive cells with all the non-edited CD90 negative cells. And similarly to what I showed you earlier for the CD34 animal, we measured in vivo editing for the CD90 transplants. We transplanted 3 of these animals, and the results are shown here in red. And overall, you can see very comparable results as compared to the CD34 animals. And again, this was done by reducing the cell number to be edited by 10-fold. I also want to point your attention to the early time point for the CD90 animal. As you can see, we initially observed a reduction in editing efficiency, and this is because we are combining the small number of CD-positive edited cells with all the non-edited CD90 negative cells. So essentially, we are diluting our editing efficiency, but we were excited to see a rebound in editing efficiency within just a week, showing that it's really the CD90 positive edited cells that are taking over and reconstituting the hematopoietic system of these animals. So, of course, we were interested in measuring fetal hemoglobin in those different animals. And without going into too much details, we basically stained blood for F cells. Those are red blood cells expressing fetal hemoglobin. And you can see in all animals a sustained increase in the frequency of F cells in the blood that was as much as 20% in frequency as compared to control animals in gray that express less than 1% of F cells. And we were also happy to see a very strong correlation between the editing efficiency that we detected in vivo and the F cell frequency in those different animals, which really established and cause and effect and validating our editing strategy for the fetal hemoglobin reactivation. So to sum up those results, we have validated the use of our refined hematopoietic stem cell phenotype for CRISPR-Cas9 editing. We have demonstrated substantial in vivo editing efficiency, and in fact, those are the highest levels that have been demonstrated so far in a large animal model. And as far as we can tell, this approach is safe. We found no adverse effects. And in experiments that I don't have time to show you, we found no off-target activity of the CRISPR-Cas9 used. And the results are now published, so you are welcome to check out the paper. And to finish, I would like to thank Hans-Peter Kim. I would like to thank members of the Kim Lab who contributed to this project, especially Stefan Rake for his expertise on the CD90 work. I would like to thank our team at the Nonhuman Primates Center, our collaborator at Seattle Children, Bluebird, and Rice University. And thank you for your attention.
Any questions? Go ahead.
Do you think this approach will actually restore the shape of the red blood cells?
So the fetal hemoglobin actually, yes, counteracts the effect of the sickling hemoglobin in sickle cell disease. And so you are going to reduce dramatically the frequency of red blood cell with a sickle shape.
Okay, great.
Any other questions?
Anything known about the biology of the repressor? Is there any safety concern about that?
So there is some safety if you are targeting the repressor directly. So if you are trying to knock out the BCL11A gene, there is concern because it is implicated in the development of B lymphocytes, for example, and is also involved in other pathways. So that's a reason why instead of targeting the BCL11A gene, we are actually targeting just the binding site, which we think is going to be safer. Mm-hmm.
So, as I understand it, you combine the CD90s with unmodified cells. Are you going to do a CD90 alone transplant? Or do you anticipate that when it goes into humans, you'll always have to kind of cover yourself by providing a backup at the same time? Because you could maintain the backup in the freezer and give the CD90s alone.
Potentially. We have been going back and forth about whether we want to do this experiment or not. We are worried about the small number of CD90 positive cells, which we are afraid, I'm not a clinician, but potentially those cells might end up in the lungs when you do a transplantation with a small cell number.
That's right. I see your point. Yeah, you're kind of filling up the space with the other cells and then minimizing the lost number of cells.
Mm-hmm. Thank you.
Okay, I think with this, like to conclude today, and like to remind you to post the session right outside. Correct? Yes, and also.